Hello, my name is Graham Farquhar. I'm at the Australian National University, and it's a pleasure to be here virtually, although I'd rather be there actually with you in Lyon. I'm going to be speaking today about carbon isotopes and carbon dioxide fixation. I have a number of interests, but that's the one that I was given today. In general, I'm interested in the mechanism of Rubisco catalysis. I'm interested in stomata and gas exchange, integration of processes underlying photosynthesis, systematic genetic and environmental variation of water requirements for plant growth, and the isotopic composition of atmospheric carbon dioxide. Plants are important, obviously, to make structures that we use for food, fuel, and fibre, and also for liberation of oxygen that we breathe through respiration. But it also requires a lot of water. The inside of a leaf is moist, and if the cells inside the leaf were exposed directly to the air, they would rapidly desiccate. Plants evolved a cuticle to cover the leaf surface and stop water loss. Plants nevertheless need atmospheric carbon dioxide. They evolved valves called stomata that allow carbon dioxide to enter and inevitably allow some water to be lost by evaporation from inside the leaf, called transpiration. Here we see the stomata on a dicotyledonous leaf with CO2 going in and water coming out. And here in a monocot, in this case it's wheat, you see the different shape of the stomata. And here's a cross-section of the leaf showing the CO2 path, which is from ambient CO2, Ca, to the intercellular space, Ci, and eventually inside the chloroplasts, which we denoted Cc. We have a very primitive way of expressing the relationship between this concentration difference and the flux A of carbon dioxide assimilation rate. We use the ratio of the CO2 flux to the CO2 gradient and call it a conductance. So that one's a stomatal conductance from here to here, and then a conductance, we call it a mesophyll conductance, to go from the intercellular spaces into the chloroplasts within the mesophyll. Hope you got all that. This is biology 101, or maybe a third year biology. Water vapor diffusion out of the leaf depends mainly on the stomatal conductance. This time it's to water vapor, and water vapor diffuses in air 1.6 times more rapidly than does carbon dioxide. And it also depends on the gradient of water vapor from this time coming out from the leaf interior to the ambient. This is the ambient humidity. So we have transpiration rate is the conductance of stomata to water times the gradient in humidity. Transpiration causes tension in the water in the plant and draws water in from the soil. That's why we need to water plants, typically about 300 molecules of water per molecule of CO2 that's fixed. Water is the greatest single limitation on plant growth. And so it raises the question about how should plants control the apertures of the stomata to assimilate the greatest amount of carbon dioxide for a given total water loss over a day, say. We treat transpiration as an expenditure and carbon dioxide assimilation as a purchase, and then take an economic approach to it. And the optimal solution is to shift the temporal pattern of expenditure of water around until no more carbon could be obtained for a given total amount of water spent over the day. At this optimum trajectory, the effect of increasing expenditure at one point of time of a small change of transpiration rate, delta E. That would require minus delta E at some other time in order to keep the total constant. When it's optimum, this will always give the same increase in A equals dE over lambda, and the same loss at the other time, dE over lambda. So that implies that the dEdA, the partial dependence of transpiration rate on assimilation rate, where the source of variation is actually stomatal conductance, should equal lambda. Lambda in this case means it's the value placed on carbon and water. So always buy more carbon if the price is less than lambda moles of water per mole of carbon. If the return on expending a marginal increase is at one part of a leaf is dE over lambda, then it should be the same for the rest of the leaf and for all the other leaves on the same plant. So always buy more carbon if the price is less than lambda mole of water per mole of carbon. Note that in dry places, water is more valuable than in moist places, and so lambda is often smaller in magnitude in dry conditions. Here's a picture of Chin Wong, who made the amazing observations of the relationship or the correlation between stomatal conductance and photosynthesis at constant temperature and humidity. This is data on Zea mays, corn, C4 plant, and each dot is an individual plant. And what we've plotted is the assimilation rate of the leaf under examination against the conductance of the stomata. And you see that with all these dots, all these plants have values that fall virtually on the same line. These lines with a larger symbol are what the plant could have chosen to do. It shows how, let's say for example with this one, what the locus would look like if stomatal conductance were to vary from 0 up to 0.3 moles per square meter per second. The plant could have chosen to be anywhere on this curve, but it chose to be here. And the choices of the plants with different amounts of nutrition or of light and so on serve to make an almost linear relationship between assimilation and conductance. And I think there's something like 100 different plants being assessed, but only 4 are examined in terms of the choice that the plant has made about stomatal conductance. And that means that if the relationship is linear through the origin, it means that the intercellular CO2 concentration is roughly constant. So that behavior that Chin Wong discovered is completely consistent with the optimization scheme of DDA being a constant in time and space. But if the plant knew exactly when the next rain was coming, it should choose a constant value of the lambda that DDA should equal, such that it would almost run out of water as the next rain arrived. But plants don't usually know. They can, however, adapt to a particular environment, soil water holding capacity, and statistics of rainfall. So between saturating rain events, lambda should decrease with time, with the time constant being the average time between events. This was shown first by Ian Cowan, then by Aniki Makela, and we've also derived some other aspects of it. Now here's some observations made by Otto Lange et al. In Israel, in the desert, and you can see how the pattern of CO2 assimilation for this species, Hamadoscoparia, changed with time as the soil dried out. Here's two other species, and you can see as the soil dry out, there develops a midday depression, and eventually almost no exchange, with a slight amount of photosynthesis in the morning and still less in the afternoon. The shape of this curve obviously reflects the leaf to air vapor pressure difference, as well as the time course of sunlight. Now, I want to make a distinction here between effective water use versus efficient water use. Optimal water use means keeping TDA constant over a day and having it decline as soil water content decreases. In an environment with frequent rain and deep soil, this means large DDA and hence large assimilation rate, large transpiration rate, large stomatal conductance, and large intercellular CO2 concentration. In other words, effectiveness here gives a low transpiration efficiency, where transpiration efficiency is defined as assimilation rate divided by the transpiration rate. However, with infrequent rain and thin soil, effectiveness leads to small values of DDA, intercellular CO2 concentration, and to high transpiration efficiency. You can imagine that in an agricultural context, where we're looking to select for certain kinds of behavior for certain climates, there would be an advantage to be able to recognize such differences across genotypes of a particular species. This is where we see a role for the stable isotopes of carbon. Diffusion of 12 CO2 through air is 1.004 4 per mil times faster than 13 CO2 through air. Now the reaction of 12 CO2 with ribulose bisphosphate, which is the first step in photosynthesis, that's catalyzed by an enzyme called Rubisco, ribulose bisphosphate carboxylase oxygenase. And the reaction of 12 CO2 is 30 per mil, 1.030 times faster than with 13 CO2, because the extra neutron makes the chemical bond harder to create or break. Carbon isotope discrimination, we expect then to be somewhere between 1.004, that's 4 per mil, and 1.030. I've defined a 12C to 13C ratio and called it Q, obviously the inverse of the normal approach where we take R as the ratio 13C to 12C, but the mathematics works out more simply this way. So if we denote the 12C 13C ratio of the plant divided by that of the atmospheric source CO2, if we denote that ratio as being 1 plus delta, where delta is called discrimination, we expect that delta is somewhere between 4 per mil and 30 per mil. The simplest description of that would be with the two endpoint linear equation, that delta is equal to 4 plus 30 minus 4 times Cc, the CO2 concentration inside the chloroplast, divided by that in the atmosphere. This is the first experimental testing of those ideas by using a range of species, including mangroves, which have a low intercellular CO2 concentration, because mangroves physiologically are in a dry environment. I say that despite the fact that they're in water, because it's brackish salt water, and it makes it expensive to get the fresh water out of it that the plant needs. And you can see that carbon isotope composition varied between plants, individual plants, where the two lines give two possible values, the values that had been recorded by biochemists for the Rubisco fractionation as being somewhere between 27 and 38 per mil. And this seemed to suggest that 27 per mil would be a good fit for the fractionation by Rubisco. Now, the assimilation rate divided by conductance is roughly proportional to CO2 assimilation rate divided by transpiration rate, as long as the genotypes that you're comparing are growing in the same conditions, the same humidity and so on. So across genotypes, discrimination should be negatively related to water use efficiency, provided any variation in the stomatal conductance to CO2 is largely driven by stomatal conductance or correlated with stomatal conductance. Here's proof empirically that transpiration efficiency, which is the water use efficiency alone with no soil water losses, so it's the dry matter production divided by the water that's transpired to make it. And here we see a number of different wheat genotypes and their water use efficiency or transpiration efficiencies plots negatively against carbon isotope discrimination. Similarly, around the same, in fact the work on wheat and peanut were about the same time, and you can see here's the result of a cross between two lines of peanut, and one parent tends to have a lower value of discrimination, that's tiftinate up here, and the other parent, Chico, which is a small, fast-growing peanut, has a high discrimination. So once again, good negative relationship. If you actually extrapolate these lines to zero transpiration efficiency, it gives a value of something like, of discrimination, something like 30 per mil, which is what the equations would predict would happen. So that zero transpiration efficiency would correspond to the stomata being totally widely open. More recently, here's some work on canola genotypes, where we plotted the other way around, carbon isotope discrimination against transpiration efficiency. This is, once again, the transpiration efficiency for production of biomass, not yield. So it's leaves and roots and shoots all lumped together. I have to point out that the step from vegetative biomass to harvested yield can be more complicated. If the end of the growing season is mild with sufficient rain and irrigation, then it pays for the plant to stretch the reproductive phase and keep on photosynthesizing and growing. If it's a dry finish, it pays for the plant to develop its seed as quickly as possible. So it turns out that with the lines of canola that we've looked at so far, lines with large discrimination tend to use the available water quickly, and so can be at an advantage when the finish is dry. So the water use efficiency of yield can have the opposite relationship with delta from the water use efficiency of biomass accumulation. This is just to show that to make the transition to commercial varieties is not a straightforward step. But in terms of genetic variation in carbon isotope discrimination and transpiration efficiency, that negative relationship among C3 plants has been reported in lots of crops and lots of trees. And working with CSIRO, this ANU technique was used to breed a wheat variety Drysdale, which was released in 2002. Since then, other lines have been released commercially. And this is the Delta team, Richard Richards, Tony Condon, Greg Rubisco, and myself in earlier days. So this is the advertising blurb that CSIRO produced for Drysdale. Drysdale is not so named because of a dry field. It's named after Russell Drysdale, an eminent Australian artist that Richard was very fond of, or is very fond of. And this is one of Drysdale's paintings that drove his wife in 1945 in the middle of a drought. Moving to the more molecular mechanistic basis of why some plants have different intercellular CO2 concentrations and therefore different discrimination. This is the first report of a particular gene affecting transpiration efficiency in Arabidopsis, and that gene is called erector. That finding seems to hold in wheat as well as Arabidopsis. The simple equation that I've been discussing thus far with two N members is obviously just an approximation to what is really much more complicated. So the current equation that we would use for detailed studies would involve ternary effects. This is the effect of water interacting with CO2, in this case 12 CO2 as well as 13 CO2. This term here is a kinetic fractionation in the boundary layer. This is the kinetic fractionation in the stomata, the 4 per mil. And this is the conductance of the mesophyll wall. And the CO2 concentrations are going ambient, the surface of the leaf, intercellular, and then to the chloroplast. Here's B, the value of about 30 per mil, which varies between species and particularly between very divergent species. And this term here relates to dark respiration continuing in the light and its fractionation. And here is the effect of photorespiratory CO2 evolution. Moving now to the isotopic composition of oxygen in water. Heavy water with an 18O or a deuterium, a hydrogen 2, has a lower vapor pressure and diffuses more slowly than the major isotopolog H2O, 1H2, 16O. And this leads to an enrichment of leaf water over source water. And the expression for the enrichment of leaf water compared to soil water depends on the fractionation between liquid and vapor. It's easier for the light molecules to go into the vapor phase. And here's the kinetic term out through the out through or in through the, depending which way you like to think of it, through the stomata. This is the enrichment of the water vapor compared to soil water, the ambient humidity and the humidity inside the leaf. It turns out that this enriched leaf water causes an enrichment in the organic oxygen of the plant and also enriches atmospheric oxygen, the Dole effect, and atmospheric carbon dioxide. Afraid we don't have time to go into much detail about that. The 18 enrichment of organic matter in wheat leaves turns out to have some practical use. These diagrams show the result of a comparison made at CIMMYT in Mexico of wheat varieties released in 1962 and up until about 1988 and the intervening years. And during that period, you can see that the stomatal conductance of these releases have gradually increased. The other thing that's noticeable is that the organic matter, O18, has decreased. Now this makes sense because as the stomatal conductance goes up, the leaves cool slightly, so that has an effect. But also it means that the kinetic isotope effect changes slightly more towards the boundary layer limitation and away from the stomatal one alone. And if we plot again here on the abscissa the grain yield, you can see that the breeders have done a good job, breeders and agronomists and so on, of improving grain yield. And that's been associated with a big increase in stomatal conductance and parenthetically in photosynthetic rate. But the organic matter, O18, is the best measure of that improvement. What is the relevance of this to geochemistry? Understanding the dependence of Rubisco fractionation on the kinetic properties of the enzyme and on the carbon dioxide concentration helps in understanding the isotopic composition of sediments. Rubisco fractionation is varied among organisms and may well have changed with evolution. I've only discussed C3 photosynthesis today. The fractionation by C4 plants and by algae with CO2 concentrating mechanisms differs again. A knowledge of plant physiology is likely to reduce the dependence on vital factors, which are still called upon in studies of, for example, corals today. I'd like to thank Dan Frost, Marie Howard Ulshoff, and the other organisers of this Goldschmidt virtual meeting. Thank you very much. And I look forward to meeting you and asking you questions about geochemistry. Thank you.